Right, maybe we can begin. So for the new ones, thank you for joining us at the EUSynBioSeminars. My name is Santiago, and we're gonna listen today to Luis Grozinger's talk. And so to talk about Luis a bit, Luis obtained his bachelor's degree in computer sciences at the University of Manchester, where he studied a lot of mathematics and the theoretical side of computing. And after that, he took interest in biology as a platform for computing. So he moved to Newcastle University to start a PhD in synthetic biology with Ángel Goñi Moreno, which was an interdisciplinary group doing synthetic biology, bioinformatics, and neuroscience. To complete his PhD, he moved to the Universidad Polytechnic de Madrid to work at the Biocomputation Lab at the Centro de Biotecnología Genómica de Plantas. He's now doing a postdoc in the Biocomputation Lab, working mostly with modeling and simulating bacterial genetic networks. So with that introduction, Luis, the floor is yours.
Okay, thank you. Hello, everyone. Thank you for the introduction. I'm Luis. Here's my photo. As we said, I'm working now in the Biocomputation Lab in the CBGP in Madrid, which is headed by Ángel, who was also my PhD supervisor. And so during my PhD, I met Hussein, who's down here in this photo, Hussein Tas, who at the time was at the CMB working with Victor de Lorenzo. And he had a lot of data for me, some experimental data. And what he was doing was taking these synthetic gene networks, he was putting them in different plasmid vectors, and then transforming those plasmid vectors into different bacterial hosts. So it was a multiple host, multiple vector library. So what he was doing when he was doing that was keeping the genetic circuit the same, but he was changing the context in which that circuit was performing. Now, basically what I mean by context, my definition of context in this context is everything that in the design stage we're not designing with, that we're either unwilling or maybe we're unable to engineer. Okay, so usually what happens, we're designing a genetic circuit, we pick genetic parts, right? So we pick a promoter, we pick an RBS, we pick a terminator, we stuff them all together in an arrangement which makes a circuit. Everything else is context, or my definition of context, right? So, for example, scarce resources, maybe RNAP or ribosomes are scarce resources, and that's part of the context because it might change without us changing the genetic components of our circuit, right? So an example is in one host or in one condition or in with one carbon source, my bacteria, in my bacteria, RNAP is super abundant, right? There's no problem. However, in another condition, in another context, RNAP is scarce. And this affects the behavior of my genetic circuit, but it's impossible to design for, because in my design stage, I haven't considered anything like that, right? So that's kind of what I mean by context. Now, when Hossein was doing this, the circuits that the synthetic gene networks that he was using were genetic logic gates. In particular, they were genetic inverters. So I want to say why. So why genetic logic and genetic logic gates are useful or interesting, and kind of motivate that a bit. So it comes from computer science. It's an analogy with logic in computer science, where we have these different logic functions. They take inputs and they produce outputs. And there's lots of different ones. One of them is this inverter function. It takes one input and produces one output. And all it does is flip the input into the output. So if the input is a 0, then it's a 1. If it's a 1, then it's a 0. Okay. And stuff like laptops, computers are made by combining these logic functions. So usually the implementation is electronic, right? So here's the symbol for an electronic component, which is an inverter. It inverts A into Q. Now, as it happens, you can also come up with a genetic implementation, a genetic circuit which implements the logic function of inversion, right? So in that case, what you do is you have an output molecule, this green one here. It's expressed, but its expression is controlled by a promoter, which is repressed by this other molecule, this blue one over here. So what happens is, if there's a lot of blue, the expression is repressed, and so you don't get a lot of green. If there's not a lot of blue, then the green is expressed freely, and there's a lot. And this is kind of what we mean by implementing genetic logic. Obviously, there's more stuff you can do in logic than just flip stuff around. So one special genetic logic function is NOR. So this is NOR. Basically, what it does is it takes two inputs and produces one output. If the inputs are both 0, then it produces an output which is 1. And in every other case, it produces an output of 0. So it also has a symbol here for the NOR gate in electronics. And it also has its own, well, this is one implementation as a genetic circuit, right? And basically the implementation is you take two NOT gates and fuse them together. So instead of the NOT gate with one promoter here with one input, you have the gate with two promoters, which each have their own individual repressors. And this thing produces a kind of NOR logic. The reason why NOR is special is this thing called universality, which is an idea from computer science or maybe mathematics. I'm not sure where the line is, right? Which says that if you give me a logic circuit, I can give you one which does the exact same thing, but only using NOR. So I can connect a bunch of NOR gates together in a way which emulates your circuit. So all I need is NOR, basically. And that's why things like cello are interested in using NOT and NOR gates, because with those two kinds of gate, or more specifically with just NOR even, they can build anything you want. That's the idea. But of course, that means you have to join together a bunch of NOR gates, which is relatively, okay, so it's easy in electronics because there's wires, right, to join them together. So it's relatively difficult to do that in synthetic biology, right? We don't have wires as per se. But we still want to join them together. So how we do that is we basically make the output of one gate, so this green thing here, be the repressor for the next gate, for the input for the next gate. And then we join them all together like that in a big chain, and we make a circuit. And if we want to say what the circuit does, we want to say, okay, so if I have this much A, how much Q do I have, right? It's a difficult question to answer, but luckily you can break that down into different questions, right? So you can look at each stage individually and answer the same question for each stage. And that's basically what Hossein did when he was making measurements of these NOT gates. He was saying for each gate that I have in my library, of which there was 20, how much repressor gives me how much output expression. Okay. And the way he did that specifically was using these two plasmids. I don't think too much detail is needed. This is basically the same workflow as Cello used to characterize their gates, right? So he has this plasmid here, which is basically the output plasmid, they call it. He has this plasmid here, which is called the input plasmid. One measures the response of YFP to the repressor, and one measures the response of YFP to the IPTG. And by combining the information that you get from measuring those two plasmids, you can say, okay, so what's the response in output expression for input expression for this specific case here? So each gate has a different R, is a different repressor, is a different promoter pair, right? So what he did, take these two plasmids, and the way he measured was using flow cytometry. So he does the flow cytometry under different conditions, different concentrations of IPTG, and he gets a distribution of YFP fluorescence, like the ones on the bottom here. Now, when you make certain assumptions, which is like that basically, so you basically go from a reasonably complicated looking thing with a few interactions to a more simplified version, you can convince yourself that really all you need is the averages of those distributions. So that's what you do. You take the averages of those distributions and you make lines. Here you have a line for the input plasmid, and here you have a line for the output plasmid. And you can further convince yourself that you can join those two lines together, and then you can get a nice curve. This time is a hill function. So you can fit a hill function for each gate, and the hill function has 4 parameters. So basically you take the fluorescence distributions that you have, and for each gate you get 4 numbers, which tell you all about that gate, the gate's performance. And Hossein did this for 7 different combinations of host and plasmid, and he did it for 20 gates. Now, with those 4 numbers, you can ask a computer to simulate a circuit for you, to build a genetic circuit in silico and see how it performs. What you can also do is ask, okay, so I have my library of 20 gates. How good is it? Like, so this was part of my thesis, was sort of measuring the quality of these libraries of genetic logic gates. The way I did it was basically said, well, if a library is good, I should be able to build complex circuits from it, right? Circuits are more complex if they're bigger. Circuits are bigger if I can join more pairs of gates together. So that's what I did. I basically used this hill function model to count the number of compatible pairs of gates in the library. So on the right-hand side is like a, I don't know, heat map of this library of 20 gates, all paired up with one another. The orange squares are where the 2 gates are compatible, where you can join them together, and the blue squares are where it's not possible. And it's not possible because of differences in the performance of the gates, right? So it's actually quite disappointing, I would say. You have 20 gates, so I mean, that's a lot of possible pairs, but you only have these few, I mean, I guess there's about 20 something compatible pairs, right? This is quite disappointing performance, I would say. But Hossein had all these 7 different contexts, and actually by adding contexts, we could improve the quality of the gate library. So that's what we did in silico, is we said, well, on the left-hand side, we're using this E.coli strain, DH5 alpha. We're only using this plasmid vector PAN. What if we added a 7 plasmids? So we can have the gates in 7 plasmids or PAN plasmids, and we can mix and match as we like. And we convert a few of these blue squares into orange squares. We get a few more compatible gates, and Therefore, theoretically, we get a better library, right? We can go further. We went even further in silico. We said, well, we have Putida as well. We have actually two strains of E. coli. We have DH5 alpha. We have this C118 lambda PIR. I'm not a microbiologist, so I don't know what that is. I just see it as three different possibilities for mixing and matching these gates, and that's what I did. And you can get to the point where the majority of these squares are orange. Basically, the majority of the gates are compatible with one another, and therefore probably you can build a more complex circuit from this library than the one that we started with. So the question, the reason that this works, the reason you get more compatibility is you add more context, is because the gates perform differently in different contexts. And using the Hill function model, you can actually take those four numbers, those four parameters for each of the gates, and you can, in silico, measure the difference between the gates. So this is what this heat map on the left-hand side is. These are basically the pairwise differences between all of the gates in the library between each different contexts. The dark areas are the more similar areas, and the lighter areas have more variability. So you can have, like, I don't know, here, E. coli corner, let's say, where all these dark squares are saying, but these gates are really similar, and it happens to be that they're all in E. coli, right? And using these, the distributions of these distances, you can answer statistical significance questions also about which contextual variable is more significant than others in producing variability. And we get results about, with p-values for, okay, so host strain has more impact than changing the backbone, or Putida is more variable than E. coli, okay, which is fairly useful. On the right hand side is plotted the Hill functions for a single gate, an example of a gate in 5 different contexts, just to show you that the performance of the gates is really variable in terms of shape, expression levels, all this kind of thing. And just to make the point that maybe it's not as straightforward as saying, well, we have more expression in E. coli than Putida, so my gate is going to express more YFP, whatever. No, it doesn't really work like that. An example about this kind of non-intuitive or non-straightforward differences between contexts that I want to discuss now is in these 5 contexts, in these gates. So these are three examples of Putida and 2 examples of E. coli with sevoplasmids. Now, the 221 plasmid is a low copy, 231 is a medium copy, and 251 is a high copy plasmid. So in E. coli, increasing the copy number of the plasmid from low to medium seems to increase the output of the gate. And that more or less makes sense to me, because if I have more copies of my gene, then my gene is going to be expressed more. That just kind of makes sense in my head. However, if we switch the context over to Putida, it's actually the case that the opposite is true. So this was confusing for me. With the low copy plasmid, we have higher, so let's say max expression, than with the high copy plasmid. And for me, that was kind of backwards. So I wanted to see if there was to find a mechanism, find a pattern, find a reason why that would be the case, which is where where you're frustrated, right? You have this Hill model, which has 4 numbers, none of which really have any biological significance or meaning. None of them have a relationship with a process that's going on inside the cell. So when you're looking for a mechanism using this Hill model, it's kind of impossible. So what I did is I went back to that simplified model that we used to come up with this Hill function idea, and added back in the the complexity of the entire plasmid. And when I was basically staring at that, I convinced myself that the only way in which I could reproduce that effect through some sort of contextual difference is if the ribosome was a scarce resource, right? So there was competition for the ribosome, by which I mean I can have lots of mRNA which is kind of queuing up for ribosome, let's say, which is sharing ribosome with all the other mRNA in the cell and from the rest of the plasmid, etc., etc. And further, I became convinced that you could required competition for ribosomes, but also that the competition was unfair. So that means that this RBS was basically stronger than all the others. So there was competition, and most of the ribosomes were getting eaten up by the repressor in the circuit. So I basically modeled all of this and simulated it. So this is what I'm modeling on the left-hand side. And first of all, I simulate it without any competition, by which I mean there's lots of ribosome is not a scarce resource. There's loads of ribosome everywhere, and it's available for everybody. This is the kind of performance you get. So as we go up in copy number from 1 to 4 to 16, we get like the intuitive thing that in my head makes sense, right? We increase the expression, that we get more copies of the genes, more expression. Okay, that's fine. Then I simulate fair competition, and again, this is the same thing. So it's the stuff that makes sense to me. As I increase the copy number, the expression rate is increasing, getting more YFP abundance. And to finally test the hypothesis, we have to model the case where the competition is unfair. And then suddenly the thing changes, and actually we have the inverse relationship where so the blue line is 1 copy, orange is 4, and the green is 16. So as the copy number increases of the plasmid, we're going down in the expression level. That was done purely as modeling, so the hypothesis is kind of still a hypothesis. We're not testing anything in experimentally yet, but for me it's compelling, and it is a hypothesis which explains this observed experimental data. So I basically concluded that there's three options. In E. coli, there's no competition for ribosome, so there's lots of abundant ribosome. But in Putida, the ribosome is scarce, and there's an unfair competition between the genes in the proteins in the circuit. Number 2, while E. coli could have scarce ribosome also, but the competition between the genes in the circuit is fair, which would mean that the RBS which is a lot stronger than another in Putida is kind of equal in E. coli. There's a difference in the ribosome binding site strength between the two contexts. And again, Putida would have to have an unfair competition for the ribosomes. Oh, the third option is that there's some of the you can't exclude some of the mechanism being responsible for the effect, right? So those are the conclusions from this example and from this kind of like modeling study, which brings me then to kind of conclusions of the talk, let's say. So the first conclusion of the talk is that the context, as we saw with those kind of heat maps where we're getting more compatible pairs as we're including different contexts, help build more complex genetic circuits. So cross-context libraries will be more powerful than single context libraries. Number 5, context interactions with circuits are not straightforward. So this thing where copy number affects a circuit in one way in one context, but in the opposite way in the other context, is not a straightforward interaction for me anyway. I can't get my head around just straight away how that would work, right? And finally, even though they're not straightforward interactions, there is hope to kind of wheedle them out. But if we stay with models like Hill functions, which are very useful for designing circuits and investigating differences between genetic logic gates, we won't be able to pick apart the details of the contextual interactions, which are actually, in practice, stopping the modularity of our genetic logic circuits. Okay, so I think that's it. I don't know how long that took, but if there's any questions, I'll be if there's any time for questions, I'd be happy to answer if I can.
Yep. Thank you very much, Luis. Absolutely. If there are any questions, there's time for that. So if anyone was to do it, I think they can unmute themselves and ask, I believe.
So I have a question. Hi, Luis. Thanks for the very nice talk, and I'm happy there is hope. So I was wondering, so you think, I mean, the different strength of the different promoter in the different contexts basically affects the final output. But if we were able to type all the various promoter in the various organisms, do you think we would be able to predict upstream the actual strength?
Okay, so
now you know if you have to do it again, you will know that promoter will be competed unfairly, right? So once it's known, it can be.
Yeah. So this is kind of so first of all, I'm not sure whether we've got crossed wires slightly. So I'm saying for this specific example, it's not the strength of the promoter which I care about. It's the strength of the RBS and the RBS, right? So I'm saying that it's this unfair competition for ribosomes which is affecting. So the transcription rate.
Sorry, I wanted to say that if you can type the RBS, I don't know why I said promoter.
Yeah. So the second part is, I mean, this applies to both cases, whether it would be transcription or translation. I'm doing like reverse engineering in this case, right? So I already have the data on the gate as it is, the experimental data, and then I'm saying, okay, so I need to find parameters for my model which reproduce this effect, right? But if I find parameters for my model in this case, they should apply, like you're kind of hinting, I guess, to the forward engineering case as well. So that in the future, I might not need to collect experimental data in lots of different contexts. I can just collect my data in one context and say, well, in E. coli, it behaved like this. But I know that this parameter changes from E. coli to Putida. So if I change that parameter, I can just in silico kind of model, simulate the effect of moving my construct from E. coli to Putida. And that's like the holy grail of this thing. The extent to which you can do that, I don't know, but we need to first be collecting lots of data in different contexts in order to do the kind of reverse engineering that I show in this example here, right? But yeah.
Yes, I had a question. Yeah, I had a question that basically follows on that thing you just said. Do you think we already have enough data on synthetic circuits and on synthetic circuits on bacteria in order to already sort of like go back steps on your simulations and then try and simulate what we already have? Or do you have the feeling that we still need to continue doing experimental analysis for you to be able to validate your models?
Yeah. No, I think we probably have enough data somewhere, like spread out, kind of. But I don't think I personally would have enough data in my lab to be able to do it. Also, I think I would like to see a bit more, less. This thing is weird to me. We're interested in measuring like YFP output for some concentration of repressor, but we have all of this other stuff in the plasmid, right? And when you're modeling, basically, if you start adding interactions and parameters and stuff like that, we end up with the problem of, they call it identifiability, parameter identifiability, which is basically like, I can't actually tell from the data. There's no way, it's stronger than that. There's no possible way that I could ever tell from the data I have what the parameter of this single parameter is, because it gets muddled up with all the different parameters, right? So I think the smaller the things that we measure, is in like the smaller the constructs that are we measure, the better we'd be off. And I think maybe I mean, when I started working on this thing, I was thinking about it in terms of this stuff up here, right? I kind of forgot all about this other bit, right? Because it wasn't interesting, let's say. But actually, it can be that kind of behind the scenes stuff is responsible for some weird behavior.
Yeah, but now that you saw this, and now you saw that the more complex the circuit is from beforehand, like from the other parts. Now, do you already have like a next idea, like a next favorite idea of things to test that maybe are like a simpler possibility for you to sort of define your parameters on the experimental data that you have, or you're still sort of like trying to wrap up this for the next steps?
I don't know. I mean, you can think of in terms of like the RBS stuff. What I haven't done, which is low hanging fruit, let's say, is just sticking to the RBS calculator. Like, okay, well, is it actually stronger or weaker or whatever like that? Which obviously doesn't full out 100% confirm the hypothesis, but it would give an idea, right? What I was interested in with, so there's two plasmids, right, for this. I'm going to go all the way back. So there's these two plasmids that we use as the input and the output for the two gates. I'd be interested in, this kind of goes against my complexity thing. I'd be interested in measuring these two in the same experiment, in the same plasmid, because right now there will be a correlation between the distribution over here of the input and the distribution of the output, which we don't get from the two separate experiments. To find the kind of correlation, we have to mix those two experiments together. So that might be a good next step for the experiment. Yeah.
Well, thank you very much. If anyone else has another question.
Thanks for the great talk, Lewis. I've got like perhaps the stupid question and then another question. So in terms of like you're seeing this like inverse, something that you don't expect in terms of like the copy number and the output, is it like really difficult to pull apart the effects of like like stress effects, like in pizza, if it's producing like proteases in response to stress overproduction from kind of a high functioning RBS? Are those kind of like side effects difficult to pull apart from the result in this context, or is it just by kind of like profiling every individual component that you could tie it down that it's like RBS based? And
yeah, so in terms of pulling apart, this is why there's a question mark. So there's three things here, and the last one is a question mark, right? I'm saying that this is one way in which this effect could be produced. Whether it's the only way, nobody knows, because there's so many of these contextual effects that I have to pick one to model. I have to pick one for my hypothesis, and then I mean that's where the experiment comes in. So if I really wanted to test this, I would have to go away and start engineering RBSs and stuff in order to really test the hypothesis. It could be that there's this, I don't know, like you said, it's a stress effect. So there's something else which is hidden from me, which is producing the effect. Just as this goes back to this parameter identifiability thing, right? In the big model that we can't see of all the contextual effects which are affecting my circuit, there's likely to be some things which are basically always hidden from our view. And I don't know, philosophically, that doesn't really matter from the forward engineering point of view. Like, it doesn't really matter to me whether whether it's RBSs or whether it's proteases or whatever it is, as long as I can predict what's going to happen, right? I don't care. That's from a SynBio point of view. From a systems biology point of view, obviously you want to break things down into and find out what those little mechanisms and stuff are. But from a SynBio forward engineering, who cares?
Yeah, no, it's really nice that you can kind of like do this sort of prediction of something really quite very complicated and still like maybe not have a full, you know, understanding. The prediction is the impressive bit. So my other question was around like about the output of the circuit and how when you change the output of the circuit, so you change it from YFP to another protein, do you still see the same effect from the same circuit design, and how much kind of like how much effect does the output have on the same sort of design?
Yeah, the answer is I can't tell you because we haven't tested with I don't know, another fluorescent protein or another repressor or anything like that. Something that probably would be useful is thinking about these circuits not the inputs and outputs of their gates not being proteins or repressors or whatever, but instead being this POPs unit, this transcription, I can't remember, polymerases per second or something. That's more useful, I think, than thinking about proteins. We don't know how fast the relationship between how fast we're transcribing here and how fast we're transcribing here. And then everything in between obviously depends on the identity of the protein that you're translating or transcribing, right? I don't know yet. We haven't tested with other stuff, but Cello builds pretty complex circuits using these NOT gates, and they only characterize them with YFP, but they still seem to work kind of with repressors in between. So between repressor molecules and YFP, it might be that the difference is small enough that we can get away with it, kind of.
Super cool. Thanks.
Thanks.
All right, Luis, do you think you have time for some more questions? Because someone from the audience, Ricardo Moreno, typed 3 questions. If you can answer some more.
And okay, let me check the chat.
So he has a question about toxicity, a question about mammalian cells, another one about like a different strain of coli.
So what particular application do you find in inducing expression of 2 or more targets within your system? I'm afraid in doing so, toxicity can be a major issue. Yeah, I mean, it is an issue. If you look at the original Cello paper, they do some measurements of toxicity, and I mean, they basically they optimize some of them out, I think, as in they don't use those particularly toxic ones. Whether there's a relationship between context and toxicity, that's an added complication, right? So maybe E. coli can handle X amount of such and such, and Putida can handle only Y. But yeah, I don't even attempt basically to model any toxicity issues. But you're right to be afraid, I guess, is the answer, because in Cello they measured some effect of toxicity. Yeah. Number 2, have you considered translating this idea into mammalian cells? With training and my knowledge at the moment, I don't have any idea of how to change the model in order to make it into a mammalian cell. The model basically has some assumptions. One is that there's only one compartment, so everything's floating around in one compartment. So I guess that might be different in mammalian cells, right? So it might be compartmentalized. I don't know if translation and transcription, they must be decoupled in mammalian cells, right? I don't know. Somebody can help me. But yeah, so the model is makes some assumptions which apply only to bacteria. I don't have any interest in translating the model into mammalian cells, if I'm being completely honest. I have enough problems with bacteria. There's enough complexity in bacteria to keep me happy for the rest of my career, I think, before starting thinking about mammals. Number 3, have you tried E. coli? There's a strain of E. coli, BL21. No. The answer is no. I wonder if Ricardo could maybe there's a reason why he suggests E. coli BL21.
Yeah, because E. coli BL21 is a specific strain of E. coli that has been modified for protein expression. So generally, it's like rule of thumb is that DH5 alpha is for transforming, and BL21 would be for expressing high concentrations of proteins.
Okay. So the idea would be that the ribosome would not be a scarce resource in BL21, whereas it might be in DH5 alpha, I guess. Yeah, that could be. Yeah, but that could be interesting. Then that could be a way to test the hypothesis.
Yep. All right. So I guess those are enough questions for you, Lewis. Thank you very much for your time and for answering all of them.
It was a pleasure.
So I guess we just can call it a day. Thank you for the talk, and thank you everyone for assisting. And Ricardo is saying I would recommend checking Jason Chin work. He's done very powerful studies in bacterial engineering, so that's something maybe you can take home, Lewis.
Okay, thank you, Ricardo.
All right. Well, thank you very much for everyone, and I guess we'll be seeing each other on the next seminars.
Thank you, everyone. Bye-bye.
Bye-bye.